have developed a technique to change patient stem cells into specialized immune cells called monocytes. Monocytes are type of cells that can change into several other cell types that affect injury and inflammation. We are investigating monocytes in the role of age-related macular degeneration, one of the most common causes of vision loss. This stepwise protocol comprises seeding of undifferentiated induced pluripotent stem cells and embryonic body formation, which is clumps of cells floating in liquid medium. After this three-dimensional culture, embryonic bodies are plated, spread out, and differentiate into monocytes. One of the advantages of this technique is that monocytes can be harvested 2 times per week for up to 4 weeks after seeding embryonic bodies. It's like a monocyte factory. Cells can be frozen and thawed later for experiments. Freshly harvested and thawed monocytes can be grown into specialized cells called M1 and M2 macrophages. One of the major functions of M1 is promoting inflammatory responses, and M2 promotes blood vessel growth. Analysis of M1 and M2 show appropriate cell shape and key cell surface markers. They also behave appropriately, ingesting lipoprotein and bacteria. Monocytes can produce not only macrophages, but also dendritic cells and microglial cells that are other important immune cells. This monocyte factory enables a reliable supply of patient-derived immune cells for therapeutic development in eye disease.